Okay, so thanks for having me. I'm really excited to be here. I love that you used to go into the Sierra, so I was happy to drive up from Irvine, although a little left. So I've been living up in the Sierra studying lakes, and I should preface this by saying not Lake Tahoe, but a bunch of different mountain lakes for the past 13 years. So I started out as a grad student in San Diego. This is my PhD advisor here. This is us in 2013. And we started sampling a suite of about 25. Irvine and Sophia was one of the field assistants one summer as well. I am originally from Nova Scotia, Canada. I'd never been above 3,000 feet in elevation before I came here, so this first summer was a shock to the system. But I'm very happy to keep working in such a beautiful place. Okay, so when you are hiking to your favorite mountain lake, if you are interested in seeing some animals on this hike, you might be thinking about birds or perhaps a merman. But the things that I'm really excited about are the millions or billions of animals that are in that lake. Little organisms called zooplankton that we're going to talk about today. And I hope I can make you just as excited to see those in the future. Okay, so what are plankton? So the general definition of plankton is living things that float in water and can't resist currents. So it's just all the tiny little living particles in water. And plankton are everywhere. So there's plankton in oceans, in lakes, but also in these smaller kind of ephemeral things like puddles, or even in your home at the bottom of your plants. There are all different types of organisms that are plankton. This is not one evolutionary line. There are animals that we'll be talking about today. There are fungi. So one of the most famous planktonic fungi around here is the chytrid fungus that can infect frogs, ultimately killing them. Oh, oops. We can talk about that later. There's also lots of plants that are plankton as well. But they're across the entire tree of life. So archaea, bacteria, eukaryotes, there are plankton representatives across the entire tree of life. Just give you a brief smattering of some important plankton. Here are some picoplankton. So pico means small. So these are bacteria and archaea. And it's hard to get photos of those, but this is bacteria in water. Got phytoplankton. You know, we have an expert in the front row here on all these different types. So phytoplankton, phyto means plant. So these are the plant plankton, and they're a really diverse group. There's lots of different morphologies, but they're also really important. So about half of the air that we're breathing right now is produced by these plants, by these phytoplankton. And they're also responsible for the great oxygenation event that allowed organisms to come onto the map. They do all sorts of things with climate. So for example, phytoplankton release these chemicals in the ocean, and they actually seed cloud formation. They have a huge impact on our climate and our Earth. And then we have the zooplankton. So zoo means animal. And this is a photo of marine zooplankton. Because I find them beautiful. And then I have the most famous plankton in the world, which is from SpongeBob. Even within the zooplankton, these are also not evolutionarily related to each other. There are many different evolutionary lineages that have become zooplankton. So there are multiple origins of this zooplanktonness. So lots of different convergent evolution of transparent organisms that are small size, that have some sort of way to filter water. So the two main groups that I'm going to talk about today are copepods and cladosporids. So on the left, these are our copepods, and these are the most dominant group of animals in the world by biomass. So the animal with the most biomass is these tiny little critters, which is pretty amazing because they're about 1 to 3 millimeters long. But they're so dominant because they're really dominant in the oceans as well as lakes. So copepods are one of the most common zooplankton in the oceans. We have cladoceans on the right here. These are 5 different genera. And what they have in common are these filtering appendages that are just kind of shoveling phytoplankton into their mouth constantly. And they're also covered in these transparent shells or carapaces. There are a bunch of really big differences between these 2 groups. One I wanted to talk through is the way that they reproduce. So these copepods, it's kind of this typical reproduction. There's males and females, and they undergo sexual reproduction. You can see the females here where they have egg sacs. So the ones that have 2 little things hanging off their back, those are eggs. But cladocerans undergo a completely different life history and a really unique one called cyclical parthenogenesis. And I'm going to walk through this because it's really fascinating and explains a lot of how zooplankton function.
How big are those ones?
They can be anywhere from half a millimeter up to the largest Daphnia around here, which are very big, can be up to like 4 millimeters. I'll show you a video of them later and you'll see how large they are.
Are these the same magnification?
These ones are the same. So I'm going to focus a lot on Daphne, which is this guy on the bottom right. And it's because these are really important grazers in lakes. They eat a lot of phytoplankton and also just small particles. You can think of them as just cows of the lakes, just grazing everything indiscriminately. And these are different for how lakes function, water transparency. So I know some of the variation, even in Lake Tahoe's transparency, has to do with these Daphnia and how abundant they are. Okay, so cyclical parthenogenesis. So during the growing season, Daphnia are all females. And they are reproducing clonally, so they're just making clones of themselves and they're pumping them out. So they create these haploid eggs, they grow into these neonates, they get released into the water, and they grow up and they're genetically identical. And this can happen really quickly. They reproduce every couple of days and they can make anywhere from 1 to 15 neonates at a time. And that's what happens all summer. But then once fall hits, temperatures are going decreasing, the Daphnia start doing something different. So they start producing a different type of haploid egg that is going to grow into a male Daphnia. So this is the first time that there'll be males in the lake. The Daphnia then start making this resting egg on their back. Then the male Daphnia are out in the lake. They have sexual reproduction. Having this genetic recombination to create this resting egg, and inside are 2 fraternal twin eggs. This Daphnia will then die, and we're left with just this resting egg. And it often settles to the bottom of the lake. And these are spectacular things. So these resting eggs can survive being dried, they can freeze, they can be eaten, go through a gut, they can survive for centuries. The oldest resting egg that's ever been hatched in a lab is 750 years old. That's the reaction. Excellent. Then, when conditions are good again, those fraternal twins will hatch out and the whole cycle will start again. So here is a video. You want to press?
Oh no, I'm looking for my cursor right now. I'm like, where is it? It's like lost in the sea of white.
What this actually looks like. So here on the bottom, there's going to be 2 Daphnia, and you can see their body is transparent, so you can see their feeding part. You can see their intestinal tract is often red or green because it's full of algae. And then its neonates are in its brood pouch back here. So this brood pouch is just kind of open to the water. And when they're old enough, they kind of just move their abdominal process out of the way, and the neonates pop out. Okay, so the way that they reproduce sets up some really interesting dynamics genetically. So when these eggs first hatch, at the beginning of the spring. So conditions are good, they're sensing that summer is coming, they'll hatch out. And remember, each one of those twins is genetically different from each other. So at the very beginning of the season, there's this huge boom in genetic diversity. Each different Daphnia is a separate genotype. And then over the course of the summer, that genetic diversity is going to decline. And that could be just random chance, some genotypes don't do well, but it's also through selection. So whatever the lake conditions are that year, the genotypes that are best suited to those conditions are going to be able to reproduce more clonally. So they're just going to clone themselves, they're going to do well and make more and more babies of that genotype. So at the end of the season, the genetic diversity is a lot lower than at the beginning of the season. And that kind of resets each year. And I kind of set this up as Daphnia only do this at the fall, and then they hatch out in the spring. But actually, they're even cooler than that because they can actually start creating these resting stages anytime that they sense something bad is coming. So they are able to sense fish. So they can sense the chemicals, pheromones from fish, or other dead Daphnia, or if there's some sort of algal toxin. Those are all things that will cause them to create these resting stages. And you can think of it as Daphnia sense something bad in the environment that their genotype is not going to do well at. So they create all this genetic diversity and just say, let's try to make a bunch of variation and see if we have some sort of clone that's going to do all those conditions. These resting stages are also really important for how zooplankton move around. So the Daphnia put a certain amount of lipids in the outside of these resting stages. And so you can end up with floaters and sinkers. So floaters, when there's a lot of lipids in the resting eggs, end up at the surface of the water, and then eventually the shore. So this is a photo. This gives you a sense of scale. This is, I went to a lake a couple years ago and found all these resting stages just on the edge. And so these, like I said, these can dry out. So these can just blow in the wind to other lakes, other ponds. Darwin actually studied the dispersal of zooplankton, and he concluded that they disperse on the feet of ducks. You can see how that would happen. So a duck is here instead of my hand, and it moves to the next pond, and so do the zooplankton go about that. The ones that have fewer lipids sink to the bottom of the lake. And the numbers that we're talking about are wild. So you can have like more than 10,000 of these resting eggs in 1 square meter of sediment on the bottom of the lake. So there's a lot of these resting eggs. And the ones that end up at the bottom, you can think of those instead of dispersing across space, those ones are going to be dispersing across time. So those ones will just wait at the bottom of the sediments and then hatch out when conditions are good. Often that's the next season, but it could be years, decades later. And it's because of that happening. Oh, that's next. Okay, so here's the photos of these resting eggs being produced. So on the back of each of these organisms, and these are not Daphnia, these are other species, you can see these resting stages being produced, and then those will fall to the bottom of the lake or float, and the rest of the zooplankton body will disintegrate. Okay, so because of how those eggs build up over time, we can do this thing called resurrection ecology, which is straight out of Jurassic. So we can actually go out into the lake and take a core of the sediments. And because sediments build up over time, this core represents a time series from the present day into the past. And we can chop this core up, that's what we're doing here in this photo, into different sections. And about 1 centimeter of sediment, at least in the lakes we studied, is approximately a decade. So if you have 30 centimeters of core, you can go back about 300 years of history. And there's all sorts of things you can do with that sediment. Lots of people look at the phytoplankton that were there, the algae that lived there, trying to understand and recreate past environments. You can also look at tree pollen. There's lots of things you can do with the sediment. But what we do is actually take out those resting eggs and hatch them. And once they're hatched, they will reproduce clonally. And so then you can do experiments on Daphnia that were whose parents were 300, 200, 100 years old, and actually started asking questions about how these organisms evolve in one location. So we're doing this in response to some of the fish stocking that have happened in the area. So trying to look at how Daphnia have what their traits were like before and after fish stocking. It's really hard to get these things to hatch, so it's going slowly. So I thought I would give you a little taste of the type of work that's been done using these resurrection techniques. This is a classic study from Lake Constance in Europe. This was actually the first resurrection ecology paper that ever came out. And Lake Constance underwent a really bad period where it was highly eutrophied. So all these nutrients got put in from agriculture. It got really green. They produced all these toxins. And so they hatched out Daphnia from before that happened and then after. And it turns out that Daphnia were actually able to evolve resistance to those toxins. So it didn't totally compensate. They were still not doing as well as they did before the toxins were there, but they were able to evolve some resistance. What are some of the ways that zooplankton actually do avoid predation? These would be some of the traits that we'll be looking for once we get ours to hatch more. So the first is morphological defenses. So when fish are absent, this is what these two species of Daphnia look like. And then in the presence of fish, they grow these helmets and really, really long spines. And that's to stop themselves from fitting in a fish's mouth. So a fish eats by creating a suction and sucking in water. And if your body is long enough that you get to stuck on the mouth part or the gill riggers, then you will not be eaten. So that's what they're doing. The other way that they like to avoid predation is through diel vertical migration. So their predators are mostly visual predators. And so during the day when the sun is out and they can be seen, zooplankton will migrate lower in the water column where it's dark and where they can't be eaten. And then at night, they'll come to the surface waters where food is more prevalent, and that's where they'll be. So there's a lot of competing pressures for where zooplankton will be in a water column, especially in this region. So UV is really high here at high elevation, and so that will actually cause them to be a little bit lower in the water column. Some of the lakes that we study are fishless, so they actually won't undergo that migration because it's really costly to go down into that cool, dark, low food environment for most of the day. You're not going to have as much energy, and so they this migration is really just in response to predator cues. I think that's one of the reasons mysids in Lake Tahoe had a big effect on zooplankton is they also do that migration. And so they are following their zooplankton prey up and down the water column. Okay, so this migration is the largest migration of animals on Earth. That's a common theme. Zooplankton are super important everywhere. So you might think of migrations, you think of birds or butterflies or these large ungulates. But indeed, it is the zooplankton moving up and down in the ocean and in lakes every day. That's the largest biomass movement. Okay, so if we scale this to a human, this is you running a 10K to eat and then running a 10K to go to bed. And you're doing it, once you scale it to your body length, at twice the speed of an Olympic runner. So these are massive undertakings by these organisms. So how do they move? A lot of them, the cladostridans that we talked about before, like I said, have these kind of filtering appendages and they bop around. One of the common terms for them is water fleas, which you can see how you would end up with that name. And then in the middle here, this is a copepod, and they move by this kind of whip-like motion of their antennae. And this is just a small copepod movement. They can jump really far, especially when they are sensing fish nearby. And if you scale that to a human, they could jump multiple basketball courts. And anyone who's tried to pick up copepods with a pipette over and over again knows how hard they are to catch. Now, Sophia about that. Okay, So I hope you agree that zooplankton are really cool, but they also really matter. So zooplankton play a really pivotal role in aquatic food webs. So a lot of that is because they're in this central position. So they're connecting what happens with primary producers, the algae, to higher trophic levels like fish. They are moving a lot of nutrients from algae to fish, but they're also really important in nutrient cycling and carbon sedimentation. So while they're filtering, for example, those daphnia that are filtering all the algae and little clay particles, end up creating fecal pellets that will actually sink to the sediments and stay there, so trapping that carbon. They're also really sensitive to lake conditions. So which species of zooplankton are in a lake at any given time, or how many are there, is really reflective of the lake environment, like pH, nutrient concentration, salinity. So they kind of be used as this indicator, holistic indicator of ecosystem health. We're going to talk through a very classic experiment in limnology to kind of highlight further the importance of zooplankton. So this is a classic study that was done in Wisconsin. And on one side of the lake, this is the kind of natural state of the lake, where there are 4 different trophic levels. So you have fish that eat fish, fish that eat zooplankton, and zooplankton that graze on algae. And in this kind of normal functioning of the lake, there are these fish that eat fish really reduce the number of planktons. So there are a lot of zooplankton on this side of the lake. And because there's a lot of zooplankton, they're really eating algae. And so to find out the importance of these trophic connections, the scientists divided this lake in half with an impenetrable curtain. And on the other side, they removed the 4th trophic level. So now on the left-hand side, you see that the planktivores, these fish really increased in abundance. They reduced the zooplankton. Now there's no zooplankton to graze the algae, and you can see how green the lake is. So this is the kind of experiment where you don't really need statistics to see the results. It is clear that these kind of cascading effects from top down can have really tremendous impacts on water. So in my lab, we work in mountain lakes across Yosemite and Indian National Forest, the Sequoia Kings Canyon. And we study zooplankton because a lot of the things that are happening in our mountain lakes are likely affecting the zooplankton, right? So we have introductions of fish, so predators of zooplankton. We know nutrients are increasing as dust reaches these lakes from the Central Valley. Temperatures are changing, salinity is changing. We have all these things that are affecting lake webs. And the other thing that is useful about studying lakes is they're often thought of as this kind of integrator or sentinel of change, or a canary in the coal mine. And that's because anything that happens on the land is also going to affect the lake. So if you have changes in the vegetation, changes in nutrient cycling, ultimately that's going to affect the water that's entering the lake, and anything in the air as well. So wildfires, for example, are also affecting the lake. So this is often one of the first places that gets affected by environmental change. So like I mentioned a couple times, we work out of the Sierra Nevada Aquatic Research Laboratory each summer, and I'm still working with John Shuren down at UC San Diego on this work. We bring a crew of researchers up each summer to experience the Sierra, and also partner with some local high school students and teachers to go out and help sample lakes. So that's our background and background on zooplankton. So I thought I'd also give you a little background on the lakes that we study. And we're going to go way back. So the Sierra, for the last 100 million years, has been in roughly the same latitude and had roughly the same elevations. So any change in this region has really been due to global climate fluctuations. And the biggest fluctuation that has affected the region in terms of the lakes that we study is the last glacial maxima. So during the cycles of glaciation, there was this wearing away of all the rock layers above the granite backland, that is the huge granite that we associate with the Sierra. This is the third largest batholith in the world, next to Patagonia and British Columbia. And these glaciers are scraping away all of these layers of rocks above that granite. And it results in these really kind of smooth, hollowed out places where those glaciers flowed. And then those high jagged peaks where the glacier, where they were above the glaciers. And in the bottom of these is often where you'll find the mountain lakes that we study. So in fact, almost all of the lakes in the Sierra today are in that last glacial maximum glacial event. So in that footprint of those glaciers. And we know that these lakes are experiencing changing climates. So here I'm showing the temperatures in July compared to the last 130 years. It's getting warmer. But we're also having changes in precipitation. And this one's a little bit different. It doesn't seem like there's this directional change in things getting generally wetter or generally drier. But what we do see is this great fluctuations. So drought and deluge. So increased variability year to year. I always say, people always ask me, like, just in terms of your data, what kind of snow year do you want? And I'm like, I want an average year. We never had an average year. Okay, so how does our lab actually study the ecosystem responses to these global change drivers? So the first way is through field surveys. And so this is using spatial comparisons. So you might think of studying a warm lake and a cold lake and saying, eventually this cold lake is going to become more similar to this warm lake. That would be a spatial comparison. There has been a ton of work done across the Sierra. The largest sampling effort was the Sierra Lakes Inventory Project, which happened in the late 90s, early 2000s. They went out and sampled 7,000 water bodies in the Sierra. They collected zooplankton from 460 some of those. And so this has been a really tremendous resource for just kind of giving context for what species are where and some of the kind of baseline information about how things are changing. Then there's long-term monitoring. This is a great place to talk about long-term monitoring because we have Lake Tahoe as kind of a shining example of that monitoring that happens in the Sierra. But this long-term monitoring is really important for detecting these small changes over time that might be kind of imperceptible otherwise. It also gives us a really great baseline for any extreme events that happen. So if we want to understand things like heat waves or wildfires and their effects on lakes, we really need to have some data before those events happen, right? So these kinds of monitoring programs can be really important about. And all the data that I'm going to be talking about today is from our monitoring program of these 25 lakes. The other thing that we do is experiments, and I'm not going to talk about any of this data today, but I'm happy to chat later. We recreate little ponds. We have 120 of these cattle tanks where we can manipulate plankton, algae, fish, and conditions to really get at mechanisms. Sometimes you see changes, but we don't know exactly why they're happening, and experiments can help us actually attribute mechanisms to those changes. So some of our recent experiments have looked at road salt and the impact on foodwebs and heat risk. Sometimes our experiments are big like this, and sometimes our experiments are small like that. Okay. All right. So I keep saying we go and sample lakes. So what does that actually mean? So we are not Lake Tahoe. We do not have research vessels and all the fancy equipment that exists here. We have a blow-up kayak that we hike. We can't really use two lakes, and we go out with a number of pieces of equipment and measure water chemistry. Many things go to biota, so the spiders plankton so fast. We wish we were that fast. Sometimes our questions involve doing some work on fish. We map these lakes. Sometimes we take cores. There's all sorts of different things we do, but that's kind of our normal suite of things we measure. Now these 25 lakes, they vary in all sorts of different ways. So we have some that are at lower elevation, that are warmer, higher nutrients. Some that are high elevation, surrounded by granite, quite cold, and kind of all combinations of those. Some have fish and some don't. So we can make those spatial comparisons. But if you live up here, you also know that there's a lot of temporal variation that happens as well. So our sampling has occurred across some of the driest and wettest years on record. And the snowpack can have a tremendous impact on how lakes function. And this is data actually from Steve Sadrow's publication. Steve Sadrow's associated with TURD. Hope some of you have gotten to meet him, he's great. And they've shown in Emerald Lake, which is one of their long-term study sites in Sequoia Kings Canyon, that in dry years, the temperature of the lake increases in the spring, reaches a high temperature, and decreases as you would expect. But in wet years, two things happen. So first, you have cold water entering the lake all throughout the summer because you have snowmelt. But also, this reflective ice doesn't allow the sun to heat up the water early in the season. And so the water gets warmer much later, and it often doesn't reach those same peak temperatures either. So snowpack is this really important variable in how lakes function. And this further highlights that. So our air temperature has increased. So over the last, this is showing 50 years here, average air temperature has increased by about 2 degrees Celsius. But if you look at lake temperature, that's each of these points here, it's much more variable. We don't see that just clean increase in temperature. And that's because of these wet and average years, those water temperatures are staying low. So we actually only see that signature of increasing temperature when we look at the drought years alone. So it takes a lot of years of data in the Sierra to really start to understand how things are changing through time. And that's what we found in our lake sampling as well. So we really don't see any directional change in water clarity, zooplankton, phytoplankton, but instead what we see is this difference between dry years and wet years. So in dry years, we have lower water transparency, higher phytoplankton biomass, more algae, higher nutrients, and lower zooplankton compared to these wet years. And so it's kind of this double whammy for water transparency. There's more nutrients, so more algae is proliferating, but also there's lower zooplankton, so they're not there to graze it either. So you just have a reduction in water clarity. And that's true across elevation as well, across all of our different types of lakes. Okay, the other aspect of our research is we're really interested in the effect of stocking the Sierra. So I talked about how these lakes were formed by glaciers scraping out these areas. And you can imagine if you just have a bunch of snowmelt after the glacier's gone, there's no way for fish to get there. So above 1,800 meters, 99% of the lakes in the Sierra were naturally fishless. And humans thought that didn't work. In the late 1800s, they started stocking lakes across the Sierra. At first, it was with mules, so putting pouches of water and trout on the side of mules. And then later moved to aircraft and just dumping fish into lakes. And researchers who have purposely been snorkeling when these kind of drops happen say about 80% of these fish split in half the second they come.
Oh my God.
So it doesn't take very many to even with that kill, it works. Why don't the guys stop? To create recreational fishing opportunities. Yeah, people like catching. Okay, so now across the Sierra, there's this kind of mosaic of fishiness. So in some lakes, you have these self-sustaining populations of trout. In others, you've had natural die-offs. That usually happens when there's not the appropriate spawning habitat. And so, one of the lakes that we study is fishless. And I assumed it had always been fishless. It's pretty shallow, it's pretty warm. But we looked at the stocking records, and they have put 2.5 million trout in that. Out there. So yeah, big fact.
Use for resources.
Increasing interest in these fish removals. So there's been a bunch of fish removals done to restore these natural conditions. And then in other areas, especially outside of the protected areas, there's continued stocking. Okay, so what impact do these fish have? So they do increase angling opportunities within mountain towns. This is an important source of revenue for some small towns, so I don't want to take away from that. But then these fish also have kind of really detrimental impacts to the native biota. So as you can imagine, they eat things in the lake, things like tadpoles affecting frog populations, zooplankton and macroinvertebrates. But what's really interesting is that their effects do not end at the lake edge. So they eat the larvae, a lot of the emerging insects. And so because there's fewer emerging insects, there's reductions in birds and bats. And because there's fewer amphibians emerging from the lakes, there's also a reduction in snake populations as well. So it really kind of rewires the entire web. Okay, so how do introduced fish affect zooplankton communities? So in the natural condition in these fishless lakes, at high elevation, where UV is really strong, this is what the zooplankton looks like. So you have these big black daphnia. So they actually produce melanin, they're melanized. And then the red are big copepods, and those have retinoids. And we think at least they produce these pigments to protect themselves from UV. The melanized is really costly to produce. It costs about 6 times as much energy as a non-melanized carapace. So it must be worth it for them to protect themselves from the UV. But at low elevation, where there's not that UV pressure, we see just small transparent zooplankton. Oh. That's weird. That should be the next one. So when you introduce fish to the lakes, at low elevation, they actually really don't change what species are there. You get the same ones. But they really have a huge impact on these high elevation lakes. So they completely eradicate these melanized daphnia and these large red copepods because they're visual predators and they're able to catch those things quite easily. The next thing we were interested in is if these introduced fish cause a trophic cascade, like that study we talked about earlier. So with that study, in our context, the natural condition would be on the left where there's no fish, and so there's lots of zooplankton eating the algae, so we have these clearer waters. And then when you add fish, do we see this reduction in zooplankton biomass and increase in algae? So if we start here, on the bottom, we have phytoplankton biomass, and we measure that by looking at the chlorophyll A concentration. We do see that, yes, fish lakes do have more algae. So, okay, that's check mark 1. But when we look at the zooplankton biomass, it's actually exactly the same with and without fish. It doesn't change. So this was really perplexing to us. So how can you have the same amount of zooplankton, but then algae is increasing? So we were thinking about it, and one thing that looking at just what biomass is there. So here are 2 lakes, they have the exact same number of daphnia. But what if in lake, the one on the right, these are reproducing and dying really quickly? So here, now we've had 8 daphnia in the same amount of time that lake has had 2. So if you just do a snapshot, you're only capturing the same biomass, but this one's much more productive. And so what we needed to do is figure out a way to actually measure the daphnia growing and reproducing. And so we found this kind of new technique where we look at this enzyme. And this is an enzyme that all zooplankton use when they are growing and reproducing, phytobiase. And when because it is a signature of growing and reproducing, when we look at these 2 lakes, we could actually now see that these ones are much more productive. Okay, so we can take lake water from all these lakes, do a bunch of chemistry, end up with fluorescent green protein that will tell us how much of that enzyme is there. So this is what I showed you before. Zooplankton biomass is the same with and without fish. But when we look at productivity, we see that yes, indeed, the fish are reducing the productivity of zooplankton. So we are seeing this trophic cascade, but it's not through changes in biomass, it's through changes in reproduction and growth rates. So some of the things that we're working on now, we're working with Yosemite on a bunch of lake restorations. They're doing continued fish removal in some areas, and we are interested in the speed of recovery of not just zooplankton, but other aspects of lake ecosystem functioning in general. And because we know that fish management and fish removal is a very controversial topic in this year, we're also doing a little bit of social science and actually doing a survey of people and how they perceive the fisheries management in this region. I would love to, if anyone would like to respond to the survey, I'll show this QR code again later. I'd love to get more and more opinions. We're kind of focusing it on how the public perceptions differ between locals and visitors, because that's kind of a distinction that we have noticed just talking. Yeah, so our lab is hoping to continue to come to these lakes for many, many more years, continue exposing our suburbanite UCI undergrads to the fantastic world that is the mountain lakes, and really start to understand what is going to be the future of this. And so with that, I'd like to thank all the people and funding sources, and also thank you for coming tonight.
The same research being done in your people?
Yeah, I think there's a lot of different overlapping questions that are happening here as well, for sure. So I was reading all sorts of stuff on like the mysids, there was fish introductions and changes in zooplankton dynamics as well.
Very interesting. Nice. In Lake Tahoe, there seems to be a kind of a competition between the daphne and the mysis. And you have any insight into the factors that affect that?
It's a great question. I know very little about mysids. They don't exist in any of our mountain lakes. So I don't have great answers for you. I was reading about it today to be like, someone's going to ask me about mysids, and I don't know enough about mysids. Does anyone in the room have an answer to that question?
What is it exactly about the mice? Is a competition between the two? I mean, they're not really competing. The mice are definitely eating them.
Yeah.
So I'm like, I don't know. That doesn't make it a competition in my brain because
Predation.
yeah.
Yeah, so it's a combination. So mysids is what I did Wednesday. So they do eat algae and detritus, but they also eat zooplankton as well. So recently, there was this decrease in mysids and a recovery of the daphnia population. So that seems to be more of a predation thing than a competition.
Hi, I have questions. I'm curious about amphibia. I don't know quite how to make this a succinct question, but I guess I'm curious, like when you go back in a sediment core, first of all, would the environmental factors that it takes to reawaken one level be like drastically different from, say, one that's like 20 centimeters higher, I'm assuming, because like they were created in different environment, maybe?
It's a great question. So the hatching cues are a bit of a black matter. I'm not going to lie,
Yeah.
including in the literature. But most of the cues are actually photoperiod related and temperature related. And so those don't seem to change back through time, but the amount of hatching that you'll get really rapidly decreases, and that's probably about the quality of the resting egg. So if it's produced in the last 10 years, you can get like 8% hatching rate. It's 100 years ago, you get like 10%. And I don't think it's hatching cues, I think it's quality of the resting eggs.
Okay. And then as also I think amphibia are super cool. So how do I sorry, I have to like think about how I want to ask this. I feel like I just blanked on it. I'll talk to you later.
So we learned in docent class that when they introduced the mysis, they are to be feedstock for introduced sport fish. And it didn't work because the fish feed during the day, and during the day the mice just go deep, and at night they come to the surface. And so I automatically thought, well, then the reason that the Daphnia are disappearing is that the Daphnia, well, we know the mice eat the Daphnia, but it turns out the Daphnia also go deep during the day and then come to the surface at night.
They do.
So I'm confused.
Well, would they go, 1,645 feet deep?
So the answer to that is no. So mysids go down into the actual benthic. So they'll go down into the sediment.
Oh,
So they go really, really deep.
they go really,
Yeah, the zooplankton just get out of the photic zone. So they're getting out of the light.
okay,
So they're not completely overlapping.
all right, okay, that makes sense, thank you. That helps.
So how did the Daphne actually clear the lake? Because that is their major role in the lake.
Yeah.
That seems to be our problem here.
Yeah.
What are they eating that's clearing the lake?
Yeah, totally. So they're eating, they're filtering, so small algae, but also little fine particles of clay and silt. Anything that is, if you have a bunch of silt that, even if it's not very much, it will like add up to reduce your salinity, right? So they're eating all of that, and then they're creating fecal pellets that are much larger than the individual silt pieces, and so because they're larger and heavier, they'll actually sink. So they're taking all the fine stuff that is clouding up the lake and making it go into the sediment.
And do those fecal pellets actually add nutrients though to the lake that cause the whole cycle to happen again?
That's a great question. So in general, in this kind of system, no. So they will go down to the sediment and remain there and kind of inert and just build up over time. So remember how I said about a centimeter of sediment is 10 years of sediment. So that is just building up over time. In other places where you have a ton of nutrients and the sediments become anoxic, so there's no more oxygen, that's when a bunch of chemistry happens and you can get recycling of phosphorus from the sediments. Don't think that happens here because there's not enough nutrients for that.
So the algae that they are feeding on is from some other nutrient coming into the lake.
Yeah, so nitrogen and phosphorus are taken up by algae, and they photosynthesize and take up carbon and create themselves, and then they get eaten. And so that is a way for nitrogen and phosphorus to get taken out of the system and put into the sediments.
This is a question I was very curious about when I came to Tahoe. In Tahoe, there's only like 4 species of zooplankton.
Which is why.
Yeah, but when I was in your lab, and I've seen in my work right now that, diversity is kind of through the roof. So do you have any insight? Like, is it the size of the system, or like what would be causing that?
It's mind boggling. I almost need to see a zooplankton and say, well, believe it. See, it's lower than most of the lakes around here. So most of the lakes around here have 7 to 12 species of zooplankton. Across the entire Sierra, there's not that many species. There's about 40 species of plankton. So it's not that diverse. But for there be only 4 here is really what? Yeah.
Yeah.
I don't.
Is it a function of the geology maybe?
Maybe, but I'm not sure why it would be different than the other smaller lakes.
Or it might be related to the introductions that happened.
I'm sure it was. Yeah, and I don't think there's zooplankton sampling that predates some of those introductions.
Where are you going with the research? Like, what's keeping you there?
It's really excited. So I think for me, I'm really interested in looking at turnover over time, trying to figure out what makes some lakes really variable year to year and what makes some lakes really stable. And both in terms of understanding Sierra lakes, but also just in a general ecological sense of what underlies biological stability. Those are the kinds of things I get excited about. But I'm also excited about kind of pairing the experimental work we do with like real world issues. So some of that like road salt work that we do is really exciting because it called like the answer actually. So yeah, doing some more applied stuff.
Where is the road salt being used? I didn't think I was really having this.
Yeah, no, so there's not a ton here. What it was is we did this globally distributed experiment. So we know that road salt is being used in a lot of places, and we want to be able to predict where it's going to have a detrimental impact on aquatic food webs and where it's not. And so we did what's called a distributed experiment where the 20 research groups across Europe and North America did the exact same experiment. And this was one of kind of the control sites where there's not a lot of road salt use. And so we expected this area to actually be really sensitive to road salt because there's not this kind of history with it. And it turns out it was actually one of the most resilient to salt. It had a very, very limited impact compared to many of the other parts of the world. And it seems like it was related to the species that were here. That was the best predictor. But yeah, no, it's a good point. Road salt is not big use here, but it's an interesting types of insights.
Well, the Daphne eat small pieces of plastic.
Yes, yeah, so there's just starting to be microplastic work done with zooplankton and more stuff in marine stuff so far, showing that copepods are eating these small pieces of plastic. We don't have any sense of what impact that has on them. So yes, but we don't know if it matters.
Are they digesting it or is it just sitting in those?
I think it's passing.
What does that do to the density? You don't think about it?
It's a good question. Yes, this is research that all needs to happen. Really.
So when we have these things, and I don't know if it's, so, we have this thing that gets in our lakes here that is very dangerous to the dogs and often kill them. Is that a is that algae? It's an algae.
Yeah.
And how does that tie into lack of zooplankton eating that algae, or do you know anything about that?
Yeah. So there are toxins that are produced by cyanobacteria, which is an algae, and they're often algae that grows like in kind of chains or clumps that produce these toxins, and so they zooplankton are both, they can die from exposure to these toxins, but also they often can't eat them because they get kind of clogged up in their filtering appendages because they're these like long strands or block things. So they do get eaten by zooplankton. That can be an important way that they the cyanobacteria decrease in abundance, but there's also many that cannot. So, and those are really driven by nutrient pollution. That's what you see active.